Our bodies are composed of millions of cells. Each one of these cells contains all the instructions detailing how our body should function. These instructions are called DNA, and they're written out in a sequence of many letters, just like a book. One complete instruction book, or genome, contains about 6 billion letters, but these are split into chapters so that our body can understand them piece by piece. One chapter of this book is what we call a gene. In other words, a gene is a particular sequence of DNA that tells our body how to carry out one particular task. But an instruction book isn't very useful on its own. The instructions must be read and carried out to make something functional. This is also true of DNA. In a process often called the central dogma, the DNA instructions are used to make proteins, which are important functional molecules. It is proteins that do things like break down the food you eat, turn sound waves into language we understand, and help cells specialize into specific materials like blood or bone. Proteins are the bedrock of almost all bodily functions. Unlike most books, every person's DNA instructions are a little bit different. This is called genetic variation. Each individual change is called a genetic variant. Most variants don't cause any functional changes at all. Imagine using the word large instead of big in a book. It doesn't alter the meaning. Some variants, though, can cause significant changes. Imagine an important word being misspelled or a whole chapter being left out. These are what we call pathogenic variants. They cause genetic disease. Pathogenic variants affect our health through the proteins that we make. If you have an instruction book with some missing information, the product you make won't be perfect. It might break easily, function poorly, or you might not be able to make enough of it. This is true of proteins too. When you make proteins based on DNA with these variants, they might be unstable, non-functional, or even completely absent. Because our body needs these proteins to function typically, this can cause genetic disorders. One such genetic disorder is called Shwachman-Diamond Syndrome, or SDS. This condition is most commonly caused by variants in a gene called SBDS. Some of these variants mean that the SBDS protein is non-functional, or that its levels are too low. For example, if the whole gene is deleted, no protein can be made. Other variants allow the SBDS protein to work, but not as well as usual. They can interfere with its function or how much of it is made. As far as we know, all individuals with SDS will have at least one mutation that allows a small amount of functional SBDS protein to be made. This is because if there was no SBDS in our body at all, we would not survive. Clearly, the SBDS protein is important for our cells and our bodies to function normally. Therefore, its loss causes some health concerns. First, people diagnosed with SDS may have some pancreatic dysfunction. One of the main jobs of the pancreas is to make important digestive enzymes. In individuals with SDS, these enzymes are not always made, probably because the cells that make them don't survive or function poorly. This can cause issues with digestion and with nutrition. Luckily, this feature of SDS is treatable by supplementing with the enzymes that are missing. So it's important for people with SDS to be monitored for this condition. Second, the SBDS protein also seems to be important for making blood cells. Without it, individuals with SDS can have low levels of different blood cell types. One of the blood cell types very commonly affected in SDS is neutrophils. When patients don't have enough neutrophils, which you might hear referred to as neutropenia, they can become more susceptible to infections. This is because neutrophils are an important part of the immune system. Other types of blood cells can be affected too. If red blood cell levels are low, individuals with SDS can develop anemia, which can cause fatigue, weakness, and dizziness. If platelet levels are low, patients' blood may not clot as well as it's supposed to. Third, in addition to affecting blood cell production, SDS can also affect the bone marrow and increase an individual's risk of blood cancers. These can begin as a condition called myelodysplastic syndrome, or MDS, which can sometimes progress to acute myeloid leukemia, or AML. It is essential to monitor patients with SDS for these changes. This is done by experienced hematologists. However, it is also important to know that not everyone with SDS will develop these conditions, and not every case of MDS will progress to leukemia. If concerning changes are found, a stem cell transplant may be recommended. If a transplant is needed, patients with SDS need a special type of regimen and benefit from consulting with SDS specialists. The way SBDS variants cause leukemia is a bit different from the way they cause other SDS features. For most genetic diseases, the genetic changes you are born with are present in all of your cells and cause symptoms directly. For example, without the SBDS protein, the pancreas may not produce enough enzymes. These changes you are born with are called germline variants because they come from your parents' eggs and sperm, and they're present in all of your cells. However, for cancer to develop, you need to accumulate other genetic changes throughout your life. These are called somatic variants, meaning they are variants present in only some cells in your body and they are not inherited from your parents. Every healthy person will gain these somatic variants as they age. That is why some people without genetic disorders can develop cancers too. However, in conditions that increase cancer risk, these genetic changes either occur more quickly or have a larger impact. That is why it is more common to develop leukemia if you have a condition like SDS. Using what we know about the genetics of SDS, we can describe how this condition is inherited. First, it is important to know that each person has 2 entire copies of the genome, the DNA instruction book, in all of their cells. One is inherited from each parent. For a lot of genes, including SBDS, you only need 1 working copy to be completely healthy. In other words, most cases of SDS are caused when the instructions inherited from both parents are non-functional or have spelling mistakes in them. This is called autosomal recessive inheritance. Mom and dad each have 1 working and 1 non-working copy, and they both pass down the non-working copy, by random chance, to their child. In other words, if both parents are what we call carriers of SDS, their children each have a 25%, or 1 in 4, chance of having SDS. If you already have a child with SDS, your future children will each have a 25% chance of inheriting the condition. Alternatively, if you yourself have SDS, 2 copies of the SBDS gene with spelling errors, and your partner is not a carrier, your children will be carriers but will not have SDS. There are a couple of exceptions to the typical autosomal recessive inheritance pattern. First, SDS is not always inherited from both parents. As mentioned previously, genetic changes are occurring in our bodies all the time. Therefore, it is possible to inherit 1 non-working copy from a parent and for the other copy to become non-functional by random chance. This is called a de novo, or new variant, because it was not inherited from either parent. The chances of this happening are very low, and it is extremely unlikely to happen multiple times within the same family. So, under these circumstances, the chance of having a second child with SDS would be extremely low. The second exception involves some of the other genes known to cause SDS or SDS-like syndromes. When the genes SBDS, EFL1, and DNA-JC21 cause SDS, or an SDS-like syndrome, they are usually inherited like the example above. However, SDS-like syndromes can also be caused by a gene called SRP54. In this case, the body needs both copies of the instructions encoded by this gene to be working in order for the body to function normally. If even one of the 2 copies has a pathogenic variant, the SDS-like syndrome will occur. This is called autosomal dominant inheritance. If a parent has this variant and their partner doesn't, there is a 50% or 1 in 2 risk of this syndrome for any of their children. Additionally, de novo occurrences of SRP54 variants, as for any gene, are possible too. Given these causes of SDS, how do we confirm a diagnosis? This is generally done through genetic testing. If a person seems to have the signs and symptoms of SDS, The first step is to take a closer look at the genes that cause the syndrome. The genes are read to look for spelling errors. To do this, a blood or saliva sample is sent to a lab, the DNA is isolated, and a machine reads the DNA sequence. To confirm an SDS diagnosis, finding just one spelling mistake in a recessive gene, like the SBDS gene, is not sufficient. Testing needs to confirm that both copies of the gene are not working. This means that there need to be 2 misspellings, one in each copy of the gene. For example, say the typical gene is supposed to be spelled the big dog. Each person would have 2 copies of that phrase. If one copy is misspelled and says the big cat, the other copy is still functional and the person would not have SDS. If one copy has 2 misspellings and says the small cat, the other copy is still functional and the person would not have SDS. However, if both copies are misspelled to the big cat, neither has the correct meaning, both copies are non-working, and SDS occurs. The easiest way to confirm that both copies are misspelled is by testing the biological parents. If both parents turn out to be carriers, that is, have one normal and one non-working copy of the gene, then it's extremely likely that each of them passed down their misspelled copy instead of their normal copy. This means the child would have 2 non-working copies and no working copies of the gene, and would therefore have SDS. If the parents are not available for testing, or if the testing is inconclusive, other techniques may be available. An experienced geneticist or SDS expert will be able to guide you through the testing strategy. This concludes our brief video on the genetics of Shwachman-Diamond syndrome. For more information and resources on all things SDS, please visit the Shwachman-Diamond Syndrome Alliance at www.sdsalliance.org. If you believe that you should get tested for SDS but have difficulties accessing testing, please reach out to us at testing@sdsalliance.org. We may be able to help you access no-cost testing or guide you to knowledgeable providers in your area. Thank you for listening.